Rift Valley fever or RVF is a viral disease or a viral hemorrhagic fever that can cause no symptoms to mild or even severe symptoms in some cases. The life cycle of Rift Valley fever involves mosquitoes, livestock, and people. The disease was first reported among livestock in the Rift Valley of Kenya in the early 1900s, and the virus was first isolated in 1931. Individuals with Rift Valley fever have no symptoms or mild illness. The latter may include fever, weakness, back pain. However, more severe symptoms, including eye disease, excessive bleeding, and swelling of the brain can occur. The severe symptom of bleeding has a probability of death of 50% in infected individuals. The virus is transmitted through mosquito vectors, as well as through contact with the tissue of infected animals. Two species of mosquitoes, Culex tritineirhynchus and Aedes vexans, are known to transmit the virus. The virus belongs to the Bunyavirales order. This is an order of enveloped negative single-stranded RNA viruses. All bunyaviruses have an outer lipid envelope with two glycoproteins, G, N, and G, C, required for cell entry. The virus G, C protein has a class II membrane fusion protein architecture similar to that found in flaviviruses and alphaviruses. The nonstructural protein encoded on the S segment, NSs, is the only component that has been found to directly affect the host. NSs is hostile and combative against the host's interferon antiviral response. IFN are essential in order for the immune system to fight off viral infections in a host. This inhibitory mechanism is believed to be due to competitive inhibition of the formation of transcription factor. On this transcription factor, NSs interacts with and binds to a subunit that is needed for RNA polymerase I and II. This interaction causes competitive inhibition with another transcription factor component and prevents the assembly process of the transcription factor complex, which results in the suppression of the host antiviral response. Diagnosis for Rift Valley fever relies on viral isolation from tissues, or serological testing with an ELISA. Other methods of diagnosis include nucleic acid testing, cell culture, and IgM antibody assays. A person's chances of becoming infected can be reduced by taking measures to decrease contact with blood, body fluids, or tissues of infected animals and protection against mosquitoes and other bloodsucking insects. Use of mosquito repellents and bed nets are two effective methods. For persons working with animals in RVF endemic areas, wearing protective equipment to avoid any exposure to blood or tissues of animals that may potentially be infected is an important protective measure. In terms of Rift Valley fever that is mild and of short duration, no treatment is required. In the case of severe Rift Valley fever, the management is general supportive therapy. RVF outbreaks occur across sub-Saharan Africa, with outbreaks occurring elsewhere infrequently. In Egypt, however, in 1977–78, an estimated 200,000 people were infected and there were at least 594 deaths. Among other notable outbreaks was a regional epidemic, in East Africa, of Rift Valley fever in late 2006 that affected Kenya, Somalia, and Tanzania. During the outbreak, 1,062 people were infected with Rift Valley fever and 394 people died between December 2006 and December 2007.